Numerous transport proteins enable plant vacuoles to accumulate metabolites and ions. The lumen of plant vacuoles is much more acidic pH 3 to 6 than is the cytosol pH 7.5. As here, the vacuolar membrane contains a V-class ATP-powered pump and a unique PPI-powered pump, both of which function to pump H+ ions into the vacuolar lumen against a concentration gradient. The vacuolar membrane also contains Cl and NO3 channels that transport these anions from the cytosol into the vacuole. Entry of these anions against their concentration gradients is driven by the inside positive potential generated by the H+ pumps. Operation of both types of proton pumps in conjunction with these anion channels produces an inside positive electric potential of about 20 mV across the vacuolar membrane and also a substantial pH gradient. The proton gradient potential across the plant vacuole membrane are used in much the same way as the Now+ gradient and electric potential across the animal cell plasma membrane, to power the selective uptake or extrusion of ions and small molecules. In the leaf, for example, excess sucrose generated during photosynthesis in the day is stored in the vacuole. During the night the stored sucrose moves into the cytoplasm and is metabolized to CO2 and H2O with concomitant generation of ATP from ADP and pi. Proton sucrose antiporter in the vacuolar membrane operates to accumulate sucrose in plant vacuoles. The inward movement is powered by the outward movement of H+, which is favored by its concentration gradient, lumen greater than cytosol, and by the outward negative potential across the vacuolar membrane uptake of Ca2+ and Now+ into the vacuole from the cytosol against their concentration gradients is similarly mediated by proton antiporters. The Now+ ATPase is also an antiport pump using the energy of ATP to pump Now+ out of the cell, K+N. An antiport pump in the vacuole of some plants harnesses the outward facilitated diffusion of protons themselves pumped into the vacuole by AH+ ATPase to the active inward transport of sodium ions. This sodium proton antiport pump enables the plant to sequester sodium ions in its vacuole. 3. Large molecules. Large molecules proteins, nucleic acids, polypeptides larger than a few amino acids, polysaccharides larger than a few sugars are not carried by transport proteins. There are mechanisms for moving larger molecules, but they don't enter into cytoplasm. 1. Mitosis is a general term for the process whereby very large particles of material are wrapped with plasma membrane and moved into the cell in the form of vesicles or vacuoles. None of the trapped material actually moves through the membrane, but remains on the other side of the original membrane, even while the vacuole is inside the cell. Thus it is defined as a process where a cell takes in macromolecules by forming vesicles derived from the plasma membrane. Vesicle forms from a localized region of plasma membrane that sinks inward, pinches off into the cytoplasm. It is used by cells to incorporate extracellular substances. There are 3 types of endocytosis. A. Pinocytosis, cell drinking. It is almost the same process as phagocytosis, except it involves liquids instead of solids. During exocytosis, a vacuole containing material to be excreted from a cell moves to the plasma membrane and fuses with it. The vacuole membrane becomes part of the plasma membrane and the contents are released to the outside. Cells use method to eliminate the wastes left after digestion and metabolism and also to release a whole variety of materials that have been synthesized inside the cell but which are needed outside the cell. Of hormones and digestive enzymes, found in multicellular animals, are 2 examples of this process. B. Phagocytosis, cell eating. Particles are engulfed by phagocytosis, a process that begins when solids make contact with the outer cell surface, triggering the movement of the membrane. Desired particles are then enclosed within a small piece of the plasma membrane which forms a sac called a vacuole, or vesicle, with the food particle inside it. This vacuole is then moved to the interior of the cell. Strictly speaking, the food particles are not yet part of the cell as it is still surrounded by membrane. Before food can be used, it must be broken down to smaller pieces and those pieces moved into the cytoplasm. Digestion occurs when the food vacuole is fused with a second vacuole, called a lysosome, that contains powerful digestive enzymes. Food is rated, its nutrients are absorbed by the cell and its waste products are left in the digestive vacuole, which may then leave the cell by exocytosis. Exocytosis occurs in the scavenging white blood cells of our body. They prowl around looking for invading bacteria and viruses which they engulf and destroy them. C. Receptor-mediated endocytosis. Endocytosis, when coated pits form vesicles when specific ligands bind to receptors on the cell's surface. It is more discriminating process than pinocytosis. These cells to acquire bulk quantities of specific substances, even if they are in low concentration in extracellular fluid, e.g. cholesterol. Membrane-embedded proteins with specific receptor sites exposed to the cell's exterior, cluster in regions called coated pits. A layer of clathrin, a fibrous protein, lines and reinforces the coated pit on the cytoplasmic side. A molecule that binds to a specific receptor site of another molecule is called a ligand. E.g. iron is carried through blood tightly bound to transferrin protein carrier. Get iron cells. Cell membrane contains special receptor proteins that bind transferrin, move towards special regions of membrane under which lie clathrin proteins. Endocytosis occurs inside clathrin, cage, moves inside cell. Cage eventually recycles back to cell surface, returning transferrin proteins to cell exterior. However, iron is released inside cell, exits from vesicles, becomes bound to ferritin. Exocytosis is the reverse of endocytosis. Quantities of material are expelled from the cell without ever passing through the membrane as individual molecules. By using the processes of endocytosis and exocytosis, some specialized types of cells move large amounts of bulk material into and out of them. It is also an active transport process. During exocytosis, a vesicle moves to the cell membrane, fuses with it, and then releases its contents to the outside of the cell. Vesicle usually budded from the Ura Golgi and migrates to plasma membrane. It is mainly used by secretory cells to export products, e.g. insulin and pancreas, or neurotransmitter from neuron. A4. Mechanism of sorting and regulation of intracellular transport. The protein biosynthetic pathways in cells can be considered to be one large sorting system. Many protein signals, usually but not always specific sequences of amino acids, that direct them to their destination, thus ensuring that they will end up in the appropriate membrane or cell compartment. These signals are a fundamental component of the sorting system. Usually the signal sequences are recognized and interact with complementary areas of proteins that serve as receptors for the proteins that contain them. Major sorting decision is made early in protein biosynthesis, when specific proteins are synthesized either on free or on membrane-bound polyribosomes. This results in two sorting branches called the cytosolic branch and the rough endoplasmic reticulum RER branch, figure 1. This sorting occurs because proteins synthesized on membrane-bound polyribosomes contain a signal peptide that mediates their attachment to the membrane of the ER. Further details on the signal peptide are given below. Proteins synthesized on free polyribosomes lack this particular signal peptide and are delivered into the cytosol. They are directed to mitochondria, nuclei, and peroxisomes by specific signals, or remain in the cytosol if they lack a signal. Any protein that contains a targeting sequence that is subsequently removed is designated as a preprotein. In some cases a second peptide is also removed, and in that event the original protein is known as a preproprotein, e.g., preproalbumin. Proteins sorted in the rough ER branch, figure 2, include many destined for various membranes, e.g., of the ER, Golgi apparatus, lysosomes, and plasma membrane, and for secretion. Lysosomal enzymes are also included. Thus, such proteins may reside in the membranes or lumens of the ER or follow the major transport route of intracellular proteins to the Golgi apparatus. Their signal-mediated sorting of certain proteins occurs in the Golgi apparatus, resulting in delivery to lysosomes, membranes of the Golgi apparatus, and other sites. Proteins destined for the plasma membrane or for secretion pass through the Golgi apparatus but generally are not thought to carry specific sorting signals. They are believed to reach their destinations by default. The entire pathway of a right-pointing arrow Golgi apparatus right-pointing arrow plasma membrane is often called the secretory or exocytotic pathway. Events along this route will be given special attention. Most reaching the Golgi apparatus or the plasma membrane are carried in transport vesicles. A brief description of the formation of these important particles will be given subsequently. Other proteins destined for secretion are carried in secretory vesicles, figure 2. These are prominent in the pancreas and certain other glands. Their mobilization and discharge are regulated and often referred to as regulated secretion, whereas the secretory pathway involving transport vesicles is called constitutive. Experimental approaches to the processes described in this chapter include 1. Use of yeast mutants, 2. Application of recombinant DNA techniques, e.g., mutating or eliminating particular sequences in proteins, or fusing new sequences onto them, and 3. Development of in vitro systems, e.g., to study translocation in the ER and mechanisms of vesicle formation. The sorting of proteins belonging to the cytosolic branch referred to above is described next, starting with mitochondrial proteins. The mitochondrion both imports and synthesizes proteins. Mitochondria contain many proteins. Their proteins, mostly membrane components of the electron transport chain, are encoded by the mitochondrial genome and synthesized in that organelle using its own protein synthesizing system. However, majority, at least several hundred, are encoded by nuclear genes, are synthesized outside the mitochondria on cytosolic polyribosomes, and must be imported. Cells have proved to be a particularly useful system for analyzing the mechanisms of import of mitochondrial proteins, partly because it has proved possible to generate a variety of mutants that have illuminated the fundamental processes involved. Most progress has been made in the study of proteins present in the mitochondrial matrix. Most progress has been made in the study of proteins present in the mitochondrial matrix, such as the F1 ATPase subunits. Only the pathway of import of matrix proteins will be discussed in any detail here. Matrix proteins must pass from cytosolic polyribosomes through the outer and inner mitochondrial membranes to reach their destination. Passage through the two membranes is called translocation. They have a pre-sequence, about 20 to 80 amino acids in length, which is not highly conserved but contains many positively charged amino acids, e.g., Li or Arg. Is equivalent to a signal peptide mediating attachment of polyribosomes to membranes of the ER, see below, but in this instance targeting proteins to the matrix. If the leader sequence is cleaved off, potential matrix proteins will not reach their destination. Translocation is believed to occur post-translationally, after the matrix proteins are released from the cytosolic polyribosomes. Interactions with a number of cytosolic proteins that act as chaperones, see below, and as targeting factors occur prior to translocation. Two distinct translocation complexes are situated in the outer and inner mitochondrial membranes, referred to respectively as TOM translocase of the outer membrane and TIM translocase of the inner membrane. Each complex analyzed and found to be composed of a number of proteins, some of which act as receptors for the incoming proteins and others as components of the transmembrane pores through which these proteins must pass. Proteins must be in the unfolded state to pass through the complexes, and this is made possible by ATP-dependent binding to several chaperone proteins. In mitochondria, they are involved in translocation, sorting, folding, assembly, and degradation of imported proteins. Photon motive force across the inner membrane is required for import. It is made up of the electric potential across the membrane inside negative and the pH gradient. The positively charged leader sequence may be helped through the membrane by the negative charge in the matrix. The pre-sequence is split off in the matrix by a matrix processing peptidase, MPP. Contact with other chaperones present in the matrix is essential to complete the overall process of import. Interaction on HSP70, HSP equals heat shock protein, ensures proper import into the matrix and prevents misfolding or aggregation, while interaction with the Mount HSP60, HSP10 system ensures proper folding. The latter proteins resemble the bacterial Bro-EL chaperonins, a subclass of chaperones that form complex cage-like assemblies made up of heptameric ring structures. The interactions of imported proteins with the above chaperones require hydrolysis of ATP to drive them. The details of how preproteins are translocated have not been fully elucidated. It's possible that the electric potential associated with the inner mitochondrial membrane causes a conformational change in the unfolded preprotein being translocated and that this helps to pull it across. Or, the fact that the matrix is more negative than the intermembrane space may attract the positively charged amino terminal of the preprotein to enter the matrix. Close contact between the membrane sites in the outer and inner membranes involved in translocation is necessary. The above describes the major pathway of proteins destined for the mitochondrial matrix. However, certain proteins insert into the outer mitochondrial membrane facilitated by the TOM complex. Others stop in the intermembrane space, and some insert into the inner membrane. Yet others proceed into the matrix and then return to the inner membrane or intermembrane space. Proteins contain two signaling sequences, one to enter the mitochondrial matrix and the other to mediate subsequent relocation, e.g., into the inner membrane. Certain mitochondrial proteins do not contain presequences, e.g., cytochrome C, which locates in the intermembrane space, and others contain internal presequences. Overall, proteins employ a variety of mechanisms and routes to attain their final destinations in mitochondria. General features that apply to the import of proteins into organelles, including mitochondria and some of the other organelles to be discussed below, are summarized in Table 1. IMPORTINS and exportins are involved in transport of macromolecules in and out of the nucleus. It has been estimated that more than 1 million macromolecules per minute are transported between the nucleus and the cytoplasm in an active eukaryotic cell. These macromolecules include histones, ribosomal proteins and ribosomal subunits, transcription factors, and mRNA molecules. The transport is bidirectional and occurs through the nuclear pore complexes, NPCs. They're complex structures with a mass approximately 30 times that of a ribosome and are composed of about 100 different proteins. The diameter of an NPC is approximately 9 nanometers but can increase up to approximately 28 nanometers. Molecules smaller than about 40 kDa can pass through the channel of the NPC by diffusion, but special translocation mechanisms exist for larger molecules. These mechanisms are under intensive investigation, but some important features have already emerged. Here we shall mainly describe nuclear import of certain macromolecules. The general picture that has emerged is that proteins to be imported, cargo molecules, carry a nuclear localization signal, NLS. One example of an NLS is the amino acid sequence Pro-2-Li-4-All-Li-Val, which is markedly rich in basic glycine residues. Depending on which NLS it contains, the cargo molecule interacts with one of a family of soluble proteins called importans, and the complex docks at the NPC. Another family of proteins called RAN plays a critical regulatory role in the interaction of the complex with the NPC and in its translocation through the NPC. RAN proteins are small monomeric nuclear GTPases and, like other GTPases, exist in either GTP-bound or GDP-bound states. They are regulated by guanine nucleotide exchange factors, GEFs, e.g., the protein RCC1 in eukaryotes, which are located in the nucleus, and RAN guanine activating proteins GAPs, which are predominantly cytoplasmic. The GTP-bound state of RAN is favored in the nucleus and the GDP-bound state in the cytoplasm. The conformations and activities of RAN molecules vary depending on whether GTP or GDP is bound to them. The GTP-bound state is active. The asymmetry between nucleus and cytoplasm, with respect to which of these two nucleotides is bound to RAN molecules, is thought to be crucial in understanding the roles of RAN in transferring complexes unidirectionally across the NPC. When cargo molecules are released inside the nucleus, the importans recirculate to the cytoplasm to be used again. Figure 3 summarizes some of the principal features in the above process. Small monomeric GPases e.g., ARF, RAB, ROS, and RO are important in various cellular processes such as vesicle formation and transport ARF and RAB. See below, certain growth and differentiation processes ROS and formation of the actin cytoskeleton. A process involving GTP and GDP is also crucial in the transport of proteins across the membrane of the ER. See below. Proteins similar to importans, referred to as exportins, are involved in export of many macromolecules from the nucleus. Cargo molecules for export carry nuclear export signals, NES. RAN proteins are involved in this process also, and it is now established that the processes of import and export share a number of common features. Most cases of Zellweger syndrome are due to mutations in genes involved in the biogenesis of peroxisomes. The peroxisome is an important organelle involved in aspects of the metabolism of many molecules, including fatty acids and other lipids, e.g., plasmalogens, cholesterol, bile acids, purines, amino acids, and hydrogen peroxide. The peroxisome is bounded by a single membrane and contains more than 50 enzymes. Catalase and urate oxidase are marker enzymes for this organelle. Its proteins are synthesized on cytosolic polyribosomes and fold prior to import. The pathways of import of a number of its proteins and enzymes have been studied, some being matrix components and others membrane components. At least 2 peroxisomal matrix targeting sequences, PTSs, have been discovered. 1. PS1 is a tripeptide, i.e., S-or-LILU, SKL, but variations of this sequence have been detected located at the carboxyl terminal of a number of matrix proteins, including catalase. Another, PS2, consisting of about 26 to 36 amino acids, has been found in at least 4 matrix proteins, e.g., thiolase, and, unlike PTS1, is cleaved after entry into the matrix. Proteins in PS1 sequences form complexes with a soluble receptor protein, PTS1R, and proteins containing PTS2 sequences complex with another, PTS2R. The resulting complexes then interact with a membrane receptor, PEX14P. Proteins involved in further transport of proteins into the matrix are also present. Most peroxisomal membrane proteins have been found to contain neither of the above 2 targeting sequences, but apparently contain others. The import system can handle intact oligomers, e.g., tetrameric catalase. Import of matrix proteins requires ATP, whereas import of membrane proteins does not. Interest in import of proteins into peroxisomes has been stimulated by studies on Zellweger syndrome. This condition is apparent at birth and is characterized by profound neurologic impairment, victims often dying within a year. Number of peroxisomes can vary from being almost normal to being virtually absent in some patients. Biochemical findings include an accumulation of very long-chain fatty acids, abnormalities of the synthesis of bile acids, and a marked reduction of plasmalogens. It to be due to mutations in genes encoding certain proteins, so-called peroxins, involved in various steps of peroxisome biogenesis, such as the import of proteins described above, or in genes encoding certain peroxisomal enzymes themselves. Two closely related conditions are neonatal adrenoleukodystrophy and infantile Riefsum disease. Zolliger syndrome and these two conditions represent a spectrum of overlapping features, with Zolliger syndrome being the most severe, many proteins affected, and infantile Riefsum disease the least severe, only one or a few proteins affected. The signal hypothesis explains how polyribosomes bind to the endoplasmic reticulum. As indicated above, the rougher branch is the second of the two branches involved in the synthesis and sorting of proteins. Proteins are synthesized on membrane-bound polyribosomes and translocated into the lumen of the rougher prior to further sorting, figure 2. The signal hypothesis was proposed by Blobel and Sabatini partly to explain the distinction between free and membrane-bound polyribosomes. They found that proteins synthesized on membrane-bound polyribosomes contain the peptide extension, signal peptide, at their amino terminals which mediated their attachment to the membranes of the ER. As noted above, proteins whose entire synthesis occurs on free polyribosomes lack this signal peptide. An important signal hypothesis was that it suggested, as turns out to be the case, that all ribosomes have the same structure and that the distinction between membrane-bound and free ribosomes depends solely on the former's carrying proteins that have signal peptides. Much evidence has confirmed the original hypothesis. Because many membrane proteins are synthesized on membrane-bound polyribosomes, the signal hypothesis plays an important role in concepts of membrane assembly. Some characteristics of signal peptides are summarized in Table 3. Figure 4 illustrates the principal features in relation to the passage of a secreted protein through the membrane of the ER. It incorporates features from the original signal hypothesis and from subsequent work. The mRNA for such a protein encodes an amino terminal signal peptide, also variously called a leader sequence, a transient insertion signal, a signal sequence, or a presequence. This hypothesis proposed that the protein is inserted into the ER membrane at the same time as its mRNA is being translated on polyribosomes, so-called co-translational insertion. As the site emerges from the large subunit of the ribosome, it is recognized by a signal recognition particle, SERP, that blocks further translation after about 70 amino acids have been polymerized, 40 buried in the large ribosomal subunit and 30 exposed. The block is referred to as elongation arrest. SERP contains 6 proteins and has a 7S RNA associated with it that is closely related to the ALU family of highly repeated DNA sequences. The SERP composed biased until the SERP signal peptide polyribosome complex has bound to the so-called docking protein, SERPR, a receptor for the SERP on the ER membrane. The SERP thus guides the signal peptide to the SERPR and prevents premature folding and expulsion of the protein being synthesized into the cytosol. The SERPR is an integral membrane protein composed of alpha and beta subunits. The alpha subunit binds GDP and the beta subunit spans the membrane. When the SERP signal peptide complex interacts with the receptor, the exchange of GDP for GTP is stimulated. Of the receptor with GTP bound has a high affinity for the SERP and thus releases the signal peptide, which binds to the translocation machinery, translocin, also present in the ER membrane. The alpha subunit then hydrolyzes its bound GTP, restoring GDP and completing a GTP-GDP cycle. The unidirectionality of this cycle helps drive the interaction of the polyribosome and its signal peptide with the ER membrane in the forward direction. The translocin consists of a number of membrane proteins that form a protein-conducting channel in the ER membrane through which the newly synthesized protein may pass. The channel appears to be open only when a signal peptide is present, preserving conductance across the ER membrane when it closes. The conductance of the channel has been measured experimentally. Specific functions of a number of components of the translocin have been identified or suggested. Translocating chain-associated membrane protein may bind the signal sequences it initially interacts with the translocin, and the Sec61P complex, consisting of 3 proteins, binds the heavy subunit of the ribosome. The insertion of the signal peptide into the conducting channel, while the other end of the parent protein is still attached to ribosomes, is termed co-translational insertion. The prolongation of the remaining portion of the protein probably facilitates passage of the nascent protein across the lipid bilayer as the ribosomes remain attached to the membrane of the ER. Thus, the rough or ribosome-studded ER is formed. It is important that the protein be kept in an unfolded state prior to entering the conducting channel, otherwise, it may not be able to gain access to the channel. Ribosomes remain attached to the ER during synthesis of signal peptide-containing proteins but are released and dissociated into their 2 types of subunits when the process is completed. Signal peptide is hydrolyzed by signal peptidase, located on the luminal side of the ER membrane, figure 4, and then is apparently rapidly degraded by proteases. Cytochrome P450, an integral ER membrane protein, does not completely cross the membrane. Instead, it resides in the membrane with its signal peptide intact. Its passage through the membrane is prevented by a sequence of amino acids called a halt or stop transfer signal. Secretory proteins and proteins destined for membranes distal to the ER completely traverse the membrane bilayer and are discharged into the lumen of the ER. N-glycan chains, if present, are added as these proteins traverse the inner part of the ER membrane, a process called co-translational glycosylation. Subsequently, the proteins are found in the lumen of the Golgi apparatus, where further changes in glycan chains occur, figure 9, prior to intracellular distribution or secretion. There is strong evidence that the signal peptide is involved in the process of protein insertion into ER membranes. Mutant proteins, containing altered signal peptides in which a hydrophobic amino acid is replaced by a hydrophilic one, are not inserted into ER membranes. Non-membrane proteins, e.g., alpha-globin, to which signal peptides have been attached by genetic engineering, can be inserted into the lumen of the ER or even secreted. There is considerable evidence that a second transposon in the ER membrane is involved in retrograde transport of various molecules from the ER lumen to the cytosol. These molecules include unfolded or misfolded glycoproteins, glycopeptides, and oligosaccharides. Some at least of these molecules are degraded in proteasome. Thus, there is two-way traffic across the air membrane. Proteins follow several routes to be inserted into or attached to the membranes of the endoplasmic reticulum. The routes that proteins follow to be inserted into the membranes of the ER include the following. A. Co-translational insertion. Figure 5 shows a variety of ways in which proteins are distributed in the plasma membrane. In particular, the amino terminals of certain proteins, e.g., the LDL receptor, can be seen to be on the extracytoplasmic face, whereas for other proteins, e.g., the agiolog-glycoprotein receptor, the carboxyl terminals are on this face. To explain these dispositions, one must consider the initial biosynthetic events at the ER membrane. The ester enters the ER membrane in a manner analogous to a secretory protein, figure 4. It partly traverses the ER membrane, its signal peptide is cleaved, and its amino terminal protrudes into the lumen. However, it is retained in the membrane because it contains a highly hydrophobic segment, the Halter-Stoph transfer signal. This sequence forms the single transmembrane segment of the protein and is its membrane anchoring domain. The small membrane in which the newly synthesized LDL receptor is located subsequently buds off as a component of a transport vesicle, probably from the transitional elements of the ER, figure 2. As described below in the discussion of asymmetry of proteins and lipids in membrane assembly, the disposition of the receptor in the ER membrane is preserved in the vesicle, which eventually fuses with the plasma membrane. In contrast, the agiolog-glycoprotein receptor possesses an internal insertion sequence, which inserts into the membrane but is not cleaved. This acts as an anchor, and its carboxyl terminal is extruded through the membrane. Complex disposition of the transporters, e.g., for glucose, can be explained by the fact that alternating transmembrane alpha helices act as uncle avenue insertion sequences and as Halt transfer signals, respectively. Each pair of helical segments is inserted as a hairpin. Sequences that determine the structure of a protein in a membrane are called topogenic sequences. As explained in the legend to figure 46-5, the above three proteins are examples of type 1, type 2, and type 3 transmembrane proteins. B. Cytosine-free polyribosomes and SUBSEQUENTATTACHMENT to the endoplasmic reticulum membrane. An example is cytochrome B5, which enters the ER membrane spontaneously. C. Retention of the luminal aspect of the ENDOPLASMICRETICULUM by specific amino acid sequences. A number of proteins possess the amino acid sequence KDEL, Li-Asp-Lulu, at their carboxyl terminal. This sequence specifies that such proteins will be attached to the inner aspect of the ER in a relatively loose manner. The chaperone BIP, see below, is one such protein. KDEL containing proteins first travel to the Golgi, interact there with a specific KDEL receptor protein, and then return in transport vesicles to the ER, where they dissociate from the receptor. D. Retrograde transport from the Golgi apparatus. Certain other non-KDEL containing proteins destined for the membranes of the ER also pass to the Golgi and then return, by retrograde vesicular transport, to the ER to be inserted therein, see below. The paragraphs demonstrate that a variety of routes are involved in assembly of the proteins of the ER membranes. A similar situation probably holds for other membranes, e.g., the mitochondrial membranes and the plasma membrane. Precise targeting sequences have been identified in some instances, e.g., KDEL sequences. Proteins move through cellular compartments to specific destinations. A scheme representing the possible flow of proteins along the ER right-pointing arrow Golgi apparatus right-pointing arrow plasma membrane route is shown in figure 6. The horizontal arrows denote transport steps that may be independent of targeting signals, whereas the vertical open arrows represent steps that depend on specific signals. Thus, flow of certain proteins, including membrane proteins, from the ER to the plasma membrane, designated bulk flow, as it is non-selective, probably occurs without any targeting sequences being involved, i.e., by default. On the other hand, insertion of resident proteins into the ER and Golgi membranes is dependent upon specific signals, e.g., KDEL or Halt transfer sequences for the ER. Transport of many enzymes to lysosomes is dependent upon the mannose-6b signal, and a signal may be involved for entry of proteins into secretory granules. Table 4 summarizes information on sequences that are known to be involved in targeting various proteins to their correct intracellular sites. Chaperones are proteins that prevent faulty folding and unproductive interactions of other proteins. Exit from the ER may be the rate-limiting step in the secretory pathway. In this context, it has been found that certain proteins play a role in the assembly or proper folding of other proteins without themselves being components of the latter. Such proteins called molecular chaperones. A number of important properties of these proteins are listed in Table 5, and the names of some of particular importance in the ER are listed in Table 6. They stabilize unfolded or partially folded intermediates, allowing them time to fold properly, and prevent inappropriate interactions, thus combating the formation of non-functional structures. Most chaperones exhibit ATPase activity and bind ADP and ATP. This activity is important for their effect on folding. The AD chaperone complex often has a high affinity for the unfolded protein, which, when bound, stimulates release of ADP. The AD chaperone complex often has a high affinity for the unfolded protein, which, when bound, stimulates release of ADP with replacement by ATP. The AT chaperone complex, in turn, releases segments of the protein that have folded properly, and the cycle involving ADP and ATP binding is repeated until the folded protein is released. Several examples of chaperones were introduced above when the sorting of mitochondrial proteins was discussed. The immunoglobulin heavy chain binding protein, BIP, is located in the lumen of the ER. This protein will bind abnormally folded immunoglobulin heavy chains and certain other proteins and prevent them from leaving the ER, in which they are degraded. Another important chaperone is calnixin, a calcium-binding protein located in the ER membrane. This protein binds a wide variety of proteins, including mixed histocompatibility (MHC) antigens and a variety of serum proteins. Calnixin binds the monoglycosylated species of glycoproteins that occur during processing of glycoproteins, retaining them in the ER until the glycoprotein has folded properly. Reticulin, which is also a calcium-binding protein, has properties similar to those of calnixin. It is not membrane-bound. Chaperones are not the only proteins in the air lumen that are concerned with proper folding of proteins. Two enzymes are present that play an active role in folding. Protein disulfide isomerase (PDI) promotes rapid reshuffling of disulfide bonds until the correct set is achieved. Peptidol isomerase (PPI) accelerates folding of proline-containing proteins by catalyzing the cis-trans isomerization of X-pro bonds, where X is any amino acid residue. Transport vesicles are key players in intracellular protein traffic. Most proteins that are synthesized on membrane-bound polyribosomes and are destined for the Golgi apparatus or plasma membrane reach these sites inside transport vesicles. The precise mechanisms by which proteins synthesized in the rough are inserted into these vesicles are not known. Though involved in transport from the air to the Golgi apparatus and vice versa, and from the Golgi to the plasma membrane, are mainly clathrin-free, unlike the coated vesicles involved in endocytosis. For the sake of clarity, the non-clathrin-coated vesicles will be referred to in this text as transport vesicles. There is evidence that proteins destined for the membranes of the Golgi apparatus contain specific signal sequences. On the other hand, most proteins destined for the plasma membrane or for secretion do not appear to contain specific signals, reaching these destinations by default. The Golgi apparatus is involved in glycosylation and sorting of proteins. The Golgi apparatus plays two important roles in membrane synthesis. First, it is involved in the processing of the oligosaccharide chains of membrane and other end-length glycoproteins and also contains enzymes involved in obligosylation. Second, it is involved in the sorting of various proteins prior to their delivery to their appropriate intracellular destinations. Parts of the Golgi apparatus participate in the first role, whereas the trans-Golgi is particularly involved in the second and is very rich in vesicles. Because of their central role in protein transport, considerable research has been conducted in recent years concerning the formation and fate of transport vesicles. A model of non-clathrin-coated vesicles involves snares and other factors. Vesicles lie at the heart of intracellular transport of many proteins. Recently, significant progress has been made in understanding the events involved in vesicle formation and transport. This has transpired because of the use of a number of approaches. These include establishment of cell-free systems with which to study vesicle formation. For instance, it is possible to observe, by electron microscopy, budding of vesicles from Golgi preparations incubated with cytosol and ATP. The development of genetic approaches for studying vesicles in yeast has also been crucial. This complex, with its own nomenclature, Table 7, and involves a variety of cytosolic and membrane proteins, GTP, ATP, and accessory factors. Based largely on a proposal by Rothman and colleagues, andrograde vesicular transport can be considered to occur in 8 steps, Figure 7. The base is that each transport vesicle bears a unique address marker consisting of one or more. The basic is that each transport vesicle bears a unique address marker consisting of one or more versus snare proteins, while each target membrane bears one or more complementary T-snare proteins with which the former interact specifically. Step 1. Coat assembly is initiated when ARF is activated by binding GTP, which is exchanged for GDP. This leads to the association of GTP-bound ARF with its putative receptor, hatched in Figure 7, in the donor membrane. Step 2. Membrane-associated ARF recruits the coat. Proteins that comprise the coat-omer shell from a cytosol, forming a coated bud. Step 3. The bud pinches off in a process involving acyl-CoA, and probably ATP, to complete the formation of the coated vesicle. Step 4. Coat disassembly involving dissociation of ARF and coat-omer shell follows hydrolysis of bound GTP. Uncoating is necessary for fusion to occur. 5. Vesicle targeting is achieved via members of a family of integral proteins, termed versus snares, that tag the vesicle during its budding. These snares pair with cognate T-snares in the target membrane to dock the vesicle. It is presumed that steps 4 and 5 are closely coupled and that step the 4th of May follows step 5, with ARF and the coat-omer shell rapidly dissociating after docking. 6. The general fusion machinery then assembles on the paired snare complex. It includes an ATPase NSF, NEM-sensitive factor and the SNAP-soluble NSF attachment factor proteins. SNAPs bind to the SNAP receptor complex, enabling NSF to bind. Step 7. Hydrolysis of ATP by NSF is essential for fusion, a process that can be inhibited by NEM-methylmelamide. Certain other proteins and calcium are also required. Step 8. Retrograde transport occurs to restart the cycle. This last step may retrieve certain proteins or recycle versus snares. Nocortisol, a microtubule disrupting agent, inhibits this step. Berfeldin A inhibits the coating process. The following points expand and clarify the above. Participate in step 1. ARF must first be modified by addition of myristic acid, C14, 0, employing myristoyl-CoA as the acyl donor. Myristoylation is one of a number of enzyme-catalyzed post-translational modifications, involving addition of certain lipids to specific residues of proteins, that facilitate the binding of proteins to the cytosolic surfaces of membranes or vesicles. Others are addition of palmitate, farnesyl, and guranylguranil. The two latter molecules are polyisoprenoids containing 15 and 20 carbon atoms, respectively. At least 3 different types of coated vesicles have been distinguished: COPI, COPII, and clathrin-coated vesicles. The first 2 are referred to here as transport vesicles. Many other types of vesicles no doubt remain to be discovered. COPI vesicles are involved in bidirectional transport from the air to the Golgi and in the reverse direction, whereas COPI vesicles are involved in bidirectional transport from the air to the Golgi and in the reverse direction, whereas COPII vesicles are involved mainly in transport in the former direction. Clathrin-containing vesicles are involved in transport from the trans-Golgi network to perolysosomes and from the plasma membrane to endosomes, respectively. Regarding selection of cargo molecules by vesicles, this appears to be primarily a function of the coat proteins of vesicles. Cargo molecules may interact with coat proteins either directly or via intermediary proteins that attach to coat proteins, and they then become enclosed in their appropriate vesicles. C. The fungal metabolite brofelvin prevents GTP from binding to ARF in step 1 and thus inhibits the entire coding process. In its presence, the Golgi apparatus appears to disintegrate, and fragments are lost. It may do this by inhibiting the guanine nucleotide exchanger involved in step D. GTP gamma S, a non-hydrolyzable analog of GTP often used in investigations of the role of GTP in biochemical processes, blocks disassembly of the coat from coated vesicles, leading to a buildup of coated vesicles. E. A family of ROS-like proteins, called the Rab protein, were required in several steps of intracellular protein transport, regulated secretion, and endocytosis. They are small monomeric GTP acids that attach to the cytosolic faces of membranes via granule-granule chains. They attach in the GTP-bound state, not shown in Figure 7, to the budding vesicle. Another family of proteins, Sec1, binds to T snares and prevents interaction with them and their complementary versus snares. When a vesicle interacts with its target membrane, Rab proteins displace Sec1 proteins and the V-snare-snare interaction is free to occur. It appears that the Rab and Sec1 families of proteins regulate the speed of vesicle formation, opposing each other. Rab proteins have been likened to throttles and Sec1 proteins to dampers on the overall process of vesicle formation. Using versus and T-snare proteins reconstituted into separate lipid bilayer vesicles have indicated that they form snare pins, i.e., snare complexes that link two membrane vesicles. F. Studies using versus and T-snare proteins reconstituted into separate lipid bilayer vesicles have indicated that they form snare pins, i.e., snare complexes that link two membrane vesicles. N and NSF are required for formation of snare pins, but once they have formed they can apparently lead to spontaneous fusion of membranes at physiologic temperature, suggesting that they are the minimal machinery required for membrane fusion. G. The fusion of synaptic vesicles with the plasma membrane of neurons involves a series of events similar to that described above. For example, 1 versus snare is designated synaptobrevin and 2 T snares are designated syntaxin and SNAP25, synaptosome-associated protein of 25 kDa. Botulinum B toxin is one of the most lethal toxins known and the most serious cause of food poisoning. This toxin is a protease that appears to cleave only synaptobrevin, thus inhibiting release of acetylcholine at the neuromuscular junction and possibly proving fatal, depending on the dose taken. Though the above model describes non-clathrin-coated vesicles, it appears likely that many of the events described above apply, at least in principle, to clathrin-coated vesicles. The assembly of membranes is complex. There are many cellular membranes, each with its own specific features. No satisfactory scheme describing the assembly of any one of these membranes is available. How various proteins are initially inserted into the membrane of the ER has been discussed above. The transport of proteins, including membrane proteins, to various parts of the cell inside vesicles has also been described. Some general points about membrane assembly remain to be addressed. Asymmetry of both proteins and lipids is maintained during membrane assembly. Vesicles from membranes of the ER and Golgi apparatus, either naturally or pinched off by homogenization, exhibit transverse asymmetries of both lipid and protein. These asymmetries are maintained during fusion of transport vesicles with the plasma membrane. The inside of the vesicles after fusion becomes the outside of the plasma membrane, and the cytoplasmic side of the vesicles remains the cytoplasmic side of the membrane, Figure 8. This asymmetry of the membranes already exists in the vesicles of the ER well before they are fused to the plasma membrane. A major problem of membrane assembly becomes understanding how the integral proteins are inserted into the lipid bilayer of the ER. Phospholipids are the major class of lipid in membranes. Enzymes responsible for the synthesis of phospholipids reside in the cytoplasmic surface of the cisternae of the ER. Synthesized at that site, they probably self-assemble into thermodynamically stable bimolecular layers, thereby expanding the membrane and perhaps promoting the detachment of so-called lipid vesicles from it. It has been proposed that these vesicles travel to other sites, donating their lipids to other membranes. However, little is known about this matter. Stated above, cytosolic proteins that take up phospholipids from one membrane and release them to another, i.e., phospholipid exchange proteins, have been demonstrated. They probably play a role in contributing to the specific lipid composition of various membranes. Lipids and proteins undergo turnover at different rates in different membranes. It has been shown that the half-lives of the lipids of the ER membranes of rat liver are generally shorter than those of its proteins, so that the turnover rates of lipids and proteins are independent. Indeed, different lipids have been found to have different half-lives. Furthermore, the half-lives of the proteins of these membranes vary quite widely, some exhibiting short hours and others long days half-lives. Thus, individual lipids and proteins of the ER membranes appear to be inserted into it relatively independently. This is the case for many other membranes. The biogenesis of membranes is thus a complex process about which much remains to be learned. One indication of the complexity involved is to consider the number of post-translational modifications that membrane proteins may be subjected to prior to attaining their mature state. The process: assembly into multimers, glycosylation, addition of a glycophosphatidylenosytol (GPI) anchor, sulfation on tyrosine or carbohydrate moieties, phosphorylation, acylation, and prenylation, a list that is undoubtedly not complete. Nevertheless, significant progress has been made. Table 8 summarizes some of the major features of membrane assembly that have emerged to date. A5. Electrical properties of membranes. Biological membranes serve as barriers to the passage of ions and polar molecules, a fact that is reflected in their high electrical resistance and capacitance. The electrical resistance is usually 10 ohm cm-2, while the capacitance is 0.5-1.5 microfarad μF cm-2. The corresponding values for artificial membranes are approximately 10 ohm cm-2 and 0.6-0.9 microfarad cm-2. The lower resistance of biological membranes must result from the presence of proteins and other ion-carrying substances or of pores in the membranes. Capacitance values for the two types of membrane are very close to those expected for a bilayer with a thickness of approximately 2.5 nanometers and a dielectric constant of 2. The electrical potential gradient is steep. surfaces usually carry a net negative charge, the result of phosphate groups of phospholipids, of carboxylate groups on proteins, and of sialic acids attached to glycoproteins. This negatively charged surface layer attracts ions of the opposite charged counterions, including protons, and repels those of the same charge. The result is development of a diffuse electrical double layer consisting of the fixed negative charges on the surface and a positive ionic atmosphere extending into the solution for a distance that depends upon the ionic strength. This ionic atmosphere is analogous to that postulated by the Debye-Huckel theory. At the focal ionic strength of 0.145 m, the thickness of the double layer, taken as the distance at which the electrical potential falls to a certain fraction of that at the cell surface, is about 0.8 nanometers. However, the double layer thickness increases to about 3 times this value at an ionic strength of 10 to 3 m and to still greater distances at lower ionic strengths. The surface charge of a cell and the associated electrical double layer are important in interactions between cells and may influence the development of extracellular structures such as basement membranes. The net negative charge on cells also gives rise to an experimentally measurable electrophoretic mobility. Characteristic of living cells is the maintenance of ionic gradients across the plasma membrane. Thus almost all cells accumulate K+, even, pumping it from very dilute external solutions. Cells also exclude sodium, pumping it out from a cytoplasm. An electrode is inserted through a cell membrane and the potential difference is measured between the inside and outside of the cell, a resting potential which, in nerve cells, may be as high as 90 mV as observed. The origin of the potential appears to lie in the concentration differences of ions. Of delta G for dilution of an ion, equation 1, and the relationship between delta G and electrode potential, equation 2, the Nernst equation 3, can be derived. A Chord equation, which applies to a single ion for which the membrane is permeable, a 10-fold concentration difference across the membrane for a monovalent ion, N equals 1, would lead to a 59 MV membrane potential, M. Since this membrane is relatively impermeable to sodium ions, it is generally conceded that for many membranes the origin of the membrane potential lies mainly with the potassium ion concentration difference which is maintained by the Na+, K+ ATPase. A more complete equation takes account of K+, Na+, and Cl, together with their respective permeabilities. Note also that equation 4 is also often called the Nernst equation. Protons are also pumped across cytoplasmic and intermitochondrial membranes. The flow of protons from inside to outside also contributes to the membrane potential. Charges of H+, K+, and other cations associated with the external membrane surface are balanced by the negative charges of protein molecules as well as Cl, and phosphate anions that are in or near to the inner surface of the membranes. Another possibility for proton flow has intrigued biophysicists for years. Membranes often display a substantial electrical conductivity in a lateral direction along the membrane surface. Electrical conduction may involve movement of protons along hydrogen-bonded lines, e.g., involving ethanolamine head groups or phosphate groups and bridging water as previously discussed. Alternatively, conduction may depend upon membrane-associated proteins. Proton conduction may be important to many proton-driven membrane processes, such as rotation of bacterial flagella, ATP synthesis, and pumping of ions. B. Structural organization and function of intracellular organelles. Cells are the structural and functional units of life. The smallest organisms are composed of only a single cell while the largest are made up of billions of cells. Even when comparing the most diverse and complex organisms, at the cellular level they are remarkably similar. Even though the human body has over 100 different cell types, they all share certain features and they even have many characteristics in common with plants. Prokaryotes to eukaryotes. It is thought that all organisms living now on Earth derive from a single primordial cell born more than 3 billion years ago. This, out-reproducing its competitors, took the lead in the process of cell division and evolution that eventually covered the Earth with green, changed the composition of its atmosphere, and made it the home of intelligent life. The family resemblances among all organisms seem too strong to be explained in any other way. Along this evolutionary road occurred about 1.5 billion years ago, when there was a transition from small cells with relatively simple internal structures, the so-called prokaryotic cells, which include the various types of bacteria, to a flourishing of larger and radically more complex eukaryotic cells such as are found in higher animals and plants. Prokaryotic cells are structurally simple but biochemically diverse. Bacteria are the simplest organisms found in most natural environments. They are spherical or rod-shaped cells, commonly several micrometers in linear dimension. They have a tough protective coat, called a cell wall, beneath which a plasma membrane encloses a single cytoplasmic compartment containing DNA, RNA, proteins, and small molecules. In the electron microscope the cell interior appears as a matrix of varying texture without any obvious organized internal structure. Bacteria are small and can replicate quickly, simply dividing in two by binary fission. When food is plentiful, survival of the fittest generally means survival of those that can divide the fastest. Under optimal conditions, a single prokaryotic cell can divide every 20 minutes and thereby give rise to 5 billion cells, approximately equal to the present human population on Earth, in less than 11 hours. The ability to divide quickly enables populations of bacteria to adapt rapidly to changes in their environment. Under lab conditions, for example, a population of bacteria maintained in a large vat will evolve within a few weeks by spontaneous mutation and natural selection to utilize new types of sugar molecules as carbon sources. Bacteria live in an enormous variety of ecological niches, and they show a corresponding richness in their underlying biochemical composition. Two distantly related can be recognized: the eubacteria, which are the commonly encountered forms that inhabit soil, water, and larger living organisms, and the archaebacteria, which are found in such incommodious environments as bogs, ocean depths, salt brines, and hot acid springs. There are bacteria that can utilize virtually any type of organic molecule as food, including sugars, amino acids, fats, hydrocarbons, polypeptides, and polysaccharides. Some even able to obtain their carbon atoms from CO2 and their nitrogen atoms from N2. Despite their relative simplicity, bacteria have existed for longer than any other organisms and still are the most abundant type of cell on Earth. Eukaryotic cells contain several distinctive organelles. As molecular oxygen accumulated in the atmosphere, what happened to the remaining anaerobic organisms with which life had begun? In a world that was rich in oxygen, which they could not use, they were at a severe disadvantage. Some, no doubt, became extinct. Others either developed the capacity for respiration or found niches in which oxygen was largely absent, where they could continue an anaerobic way of life. Others became predators or parasites on aerobic cells. And some hit upon a strategy for survival more cunning and vastly richer in implications for the future. They are believed to have formed an intimate association with an aerobic type of cell, living with it in symbiosis. This is the most plausible explanation for the metabolic organization of present-day cells of the eukaryotic type with which this book will be chiefly concerned. Eukaryotic cells, by definition and in contrast to prokaryotic cells, have a nucleus karyon in Greek which contains most of the cell's DNA, enclosed by a double layer of membrane. The DNA is kept in a compartment separate from the rest of the contents of the cell, the cytoplasm, where most of the cell's metabolic reactions occur. In the cytoplasm, moreover, many distinctive organelles can be recognized. Prominent among these are two types of small bodies, the chloroplasts and mitochondria. Each of these is enclosed in its own double layer of membrane, which is chemically different from the membrane surrounding the nucleus. Mitochondria are an almost universal feature of eukaryotic cells, whereas chloroplasts are found only in those eukaryotic cells that are capable of photosynthesis, that is, in plants but not in animals or fungi. Both organelles almost certainly have a symbiotic origin. B1. Cell wall. A wall is a fairly rigid layer surrounding a cell, located external to the cell membrane, which provides the cell with structural support, protection, and acts as a filtering mechanism. The cell wall also prevents overexpansion when water enters the cell. They are found in plants, bacteria, fungi, algae, and some archaea. Animals and protozoa do not have cell walls. The materials in a cell wall varies between species. In plants, the strongest component of the complex cell wall is a carbohydrate polymer called cellulose. In bacteria, peptidoglycan forms the cell wall. Archaean cell walls have various compositions, and may be formed of glycoprotein slayers, pseudopeptidoglycan, or polysaccharides. Fungi possess cell walls of chitin, and algae typically possess walls constructed of glycoproteins and polysaccharides, however certain algal species may have a cell wall composed of silicic acid. Often, other accessory molecules are found anchored to the cell wall. Properties. The cell wall serves a similar purpose in those organisms that possess them. The wall gives cells rigidity and strength, offering protection against mechanical stress. In multicellular organisms, it permits the organism to build and hold its shape morphogenesis. The cell wall also limits the entry of large molecules that may be toxic to the cell. It further permits the creation of a stable osmotic environment by preventing osmotic lysis and helping to retain water. The composition, properties, and form of the cell wall may change during the cell cycle and depend on growth conditions. Rigidity. In most cells, the cell wall is semi-rigid, meaning that it will bend somewhat rather than holding a fixed shape. This flexibility is seen when plants wilt, so that the stems and leaves begin to droop, or in seaweeds that bend in water currents. Wall rigidity seen in healthy plants results from a combination of the wall construction and turgor pressure. The rigidity of the cell wall thus results in part from inflation of the cell contained. This inflation is a result of the passive uptake of water. Other cell walls are inflexible. In plants, a secondary cell wall is a thicker additional layer of cellulose. Additional layers may be formed containing lignin in xylem cell walls, or containing suberin in cork cell walls. These compounds are rigid and waterproof, making the secondary wall stiff. Both wood and bark cells of trees have secondary walls. Other parts of plants such as the leaf stalk may acquire similar reinforcement to resist the strain of physical forces. Certain single-cell protists and algae also produce a rigid wall. Diatoms build a frustool from silica extracted from the surrounding water. Radiolarians also produce a test from minerals. Many green algae, such as the dicyclodales, encase their cells in a secreted skeleton of calcium carbonate. In each case, the wall is rigid and essentially inorganic. Permeability. The primary cell wall of most plant cells is semi-permeable and permit the passage of small molecules and small proteins, with size exclusion estimated to be 30 to 60 kDa. Key nutrients, especially water and carbon dioxide, are distributed throughout the plant from cell wall to cell wall in apoplastic flow. Plant cell walls. Composition. The major carbohydrates making up the primary growing cell wall are cellulose, hemicellulose and pectin. The cellulose microfibrils are linked via hemicellulosic tethers to form the cellulose-hemicellulose network, which is embedded in the pectin matrix. The most common hemicellulose in the primary cell wall is xyloglucan. Grass walls. Xyloglucan and pectin are reduced in abundance and partially replaced by glucuronorabinoxylin, a hemicellulose. Primary cells characteristically grow by a mechanism called acid growth, which involves turgor-driven movement of the strong cellulose microfibrils within the weaker hemicellulose pectin matrix, catalyzed by expansin proteins. Major polymers that make up wood, largely secondary cell walls, include cellulose 35 to 50%, xylin, a type of hemicellulose, 20 to 35%, and a complex phenolic polymer called lignin, 10 to 25%. Lignin penetrates the spaces in the cell between cellulose, hemicellulose and pectin components, driving out water and strengthening the wall. Secondary walls, especially in grasses, may also contain microscopic silicate crystals, which may strengthen the wall and protect it from herbivores. Cells walls also contain numerous enzymes, such as hydrolases, esterases, peroxidases, and transglycosylases, that cut, trim and cross-link wall polymers. Smaller than 5% of structural proteins are found in most plant cell walls. They are classified as hydroxyproline-rich glycoproteins (HRGP) or abinogalactan proteins (AGP), glycine-rich proteins (GRPs), and proline-rich proteins (PRPs). Each class of glycoprotein is defined by a characteristic, highly repetitive protein sequence. Most are glycosylated, contain hydroxyproline (HIP), and become cross-linked in the cell wall. These proteins are often concentrated in specialized cells and in cell corners. Cell walls of the epidermis and endodermis may also contain suberin or cutin, two polyester-like polymers that protect the cell from herbivores. The relative composition of carbohydrates, secondary compounds and protein varies between plants and between the cell type and age. Up to 3 straighter layers may be found in plant cell walls. The middle lamella, a layer rich in pectins. This outermost layer forming the interface between adjacent plant cells and glues them together. The primary cell wall, generally a thin, flexible and extensible layer formed while the cell is growing. The secondary cell wall, a thick layer formed inside the primary cell wall after the cell is fully grown. It is not found in all cell types. In some cells, such as found xylem, the secondary wall contains lignin, which strengthens and waterproofs the wall. Cell walls in some plant tissues also function as storage depots for carbohydrates that can be broken down and resorbed to supply the metabolic and growth needs of the plant. For example, sperm cell walls in the seeds of cereal grasses, nasturtium, and other species, were rich in glucans and other polysaccharides that are readily digested by enzymes during seed germination to form simple sugars that nourish a growing embryo. Cellulose microfibrils are not readily digested by plants, however. The middle lamella is laid down first, formed from the cell plate during cytokinesis, and the primary cell wall is then deposited inside the middle lamella. The actual structure of the cell wall is not clearly defined and several models exist: the covalently linked cross model, the tether model, the diffuse layer model and the stratified layer model. However, the primary cell wall can be defined as composed of cellulose microfibrils aligned at all angles. Microfibrils are held together by hydrogen bonds to provide a high tensile strength. This is held together and shared a gelatinous membrane called the middle lamella, which contains magnesium and calcium pectates, salts of pectic acid. Cells interact through plasmodesma (Ta), which are interconnecting channels of cytoplasm that connect to the protoplasts of adjacent cells across the cell wall. In some plants and cell types, after a maximum size or point in development has been reached, a secondary wall is constructed between the plant cell and primary wall. Unlike the primary wall, the microfibrils are aligned mostly in the same direction, and with each additional layer the orientation changes slightly. Cells with secondary cell walls are rigid. Cell-to-cell communication is possible through pits in the secondary cell wall that allow plasmodesma to connect cells through the secondary cell walls. Trees modify cell walls in their branches to reinforce and support structure. Conifers, such as pine, produce thicker cell walls on the undersides of branches to push their branches upwards. The resulting wood is called compression wood. By contrast, hardwood trees reinforce the walls on the upper sides of branches to pull their branches up. This is known as tension wood. Additional thickening may occur in other parts of the plant in response to mechanical stress. 2. Algal cell walls. Like plants, algae have cell walls. Algal cell walls contain cellulose and a variety of glycoproteins. The inclusion of additional polysaccharides in algal cells walls is used as a feature for algal taxonomy. Monoform microfibrils in the cell walls of a number of marine green algae including those from the genera, Codium, Dicycladus, and Acetabularia as well as in the walls of some red algae, like Porphyra and Bongia. Silanes. Alginic acid is a common polysaccharide in the cell walls of brown algae. Sulfoid polysaccharides occur in the cell walls of most algae. Those common in red algae include agarose, carrageenan, porphyrin, percelerin and phonorin. Other compounds that may accumulate in algal cell walls include sporopollenin and calcium ions. Known as the diatoms synthesize their cell walls, also known as frustules or valves, from silicic acid, specifically orthosilicic acid, H4CO4. The acid is polymerized intracellularly, then the wall is extruded to protect the cell. Significant relative to the organic cell walls produced by other groups, silica frustules require less energy to synthesize, approximately 8%, potentially a major saving on the overall cell energy budget and possibly an explanation for higher growth rates in diatoms. 3. Fungal cell walls. There are several groups of organisms that may be called fungi. Some of these groups have been transferred out of the kingdom fungi, in part because of fundamental biochemical differences in the composition of the cell wall. Most true fungi have a cell wall consisting largely of chitin and other polysaccharides. 8. True fungi do not have cellulose in their cell walls, but some fungus-like organisms do. 2. Chemical structure of a unit from a chitin polymer chain. True fungi. Not all species of fungi have cell walls, but in those that do, the plasma membrane is followed by 3 layers of cell wall material. From inside out these are: a chitin layer, polymer consisting mainly of unbranched chains of N-acetyl D-glucosamine. A layer of beta-1,3-glucan. A layer of monoproteins, mannose containing glycoproteins, which are heavily glycosylated at the outside of the cell. Fungus-like protists. The group Blumycetes, also known as water molds, are sa-protrophic plant pathogens like fungi. Until recently they were widely believed to be fungi, but structural and molecular evidence has led to their reclassification as heteroconts, related to autotrophic brown algae and diatoms. Unlike fungi, Blumycetes typically possess cell walls of cellulose and glucans rather than chitin, although some genera, such as Achlea and Cyprolenia, do have chitin in their walls. The fraction of cellulose in the walls is no more than 4 to 20%, far less than the fraction comprised by glucans. Blumycete cell walls also contain the amino acid hydroxyproline, which is not found in fungal cell walls. The Dictyosteles are another group formerly classified among the fungi. They are slime molds that feed as unicellular amoebae, but aggregate into a reproductive stalk in sporangium under certain conditions. Cells of the reproductive stalk, as well as the spores formed at the apex, possess a cellulose wall. This wall has been shown to possess 3 layers, the middle of which is composed primarily of cellulose, and the innermost is sensitive to cellulase and prenase. IV. Prokaryotic cell walls. Bacterial cell walls. Around the outside of the cell membrane is the bacterial cell wall. Bacterial cell walls are made of peptidoglycan, also called murin, which is made from polysaccharide chains cross-linked by unusual peptides containing D-amino acids. Bacterial cell walls are different from the cell walls of plants and fungi, which are made of cellulose and chitin, respectively. The cell wall of bacteria is also distinct from that of archaea, which do not contain peptidoglycan. The cell wall is essential to the survival of many bacteria. The antibiotic penicillin is able to kill bacteria by inhibiting a step in the synthesis of peptidoglycan. There are broadly speaking two different types of cell wall in bacteria, called gram-positive and gram-negative. The names originate from the reaction of cells to the Gram stain, a test long employed for the classification of bacterial species. Gram-positive bacteria possess a thick cell wall containing many layers of peptidoglycan and teichoic acids. Gram-negative bacteria have a relatively thin cell wall consisting of a few layers of peptidoglycan surrounded by a second lipid membrane containing lipopolysaccharides and lipoproteins. Most of the gram-negative cell wall and only the Firmicutes and actinobacteria, previously known as the low G plus C and high G plus C gram-positive bacteria, respectively, have the alternative gram-positive arrangement. These differences can produce differences in antibiotic susceptibility. For instance, vancomycin can kill only gram-positive bacteria and is ineffective against gram-negative pathogens, such as Haemophilus influenzae or Pseudomonas aeruginosa. Archaeal cell walls. Although not truly unique, the cell walls of archaea are unusual. Peptidoglycan is a standard component of all bacterial cell walls. All archaeal cell walls lack peptidoglycan, with the exception of one group of methanogens. In the group, the peptidoglycan is a modified form very different from the kind found in bacteria. There are 4 types of cell wall currently known among the archaea. One type of archaeal cell wall is that composed of pseudopeptidoglycan, also called pseudomurine. This type of wall is found in some methanogens, such as Methanobacterium and Methanodermis. While the overall structure of archaeal pseudopeptidoglycan superficially resembles that of bacterial peptidoglycan, there are a number of significant chemical differences. Like the peptidoglycan found in bacterial cell walls, pseudopeptidoglycan consists of polymer chains of glycan cross-linked by short peptide connections. However, unlike peptidoglycan, the sugar N-acetylmuramic acid is replaced by N-acetyltulosimiuronic acid, and the two sugars are bonded with a beta-1,3 glycosidic linkage instead of beta-1,4. Additionally, the cross-linking peptides are L-amino acids rather than D-amino acids as they are in bacteria. A second type of archaeal cell wall is found in Methanosarcina and Halococcus. This type of cell wall is composed entirely of a thick layer of polysaccharides, which may be sulfated in the case of Halococcus. Structure in this type of wall is complex and as yet is not fully investigated. A third type of wall among the archaea consists of glycoprotein, and occurs in the hyperthermophiles, Halobacterium, and some methanogens. In Halobacterium, the proteins in the wall have a high content of acidic amino acids, giving the wall an overall negative charge. The result is an unstable structure that is stabilized by the presence of large quantities of positive sodium ions that neutralize the charge. Consequently, Halobacterium thrives only under conditions with high salinity. In archaea, such as Methanomicrobium and Desulfurococcus, the wall may be composed only of surface layer proteins, known as an S layer. Layers are common in bacteria, where they serve as either the sole cell wall component or an outer layer in conjunction with peptidoglycan and murin. Most archaea are gram-negative, though at least one gram-positive member is known. B2. The nucleus. The presence of a nucleus is the principal feature that distinguishes eukaryotic from prokaryotic cells. By housing the cell's genome, the nucleus serves both as the repository of genetic information and as the cell's control center. DNA replication, transcription, and RNA processing all take place within the nucleus, with only the final stage of gene expression, translation, localized to the cytoplasm. By separating the genome from the cytoplasm, the nuclear envelope allows gene expression to be regulated by mechanisms that are unique to eukaryotes. Whereas mRNAs are translated while their transcription is still in process, eukaryotic mRNAs undergo post-transcriptional processing, e.g., splicing before being transported from the nucleus to the cytoplasm. The presence of the nucleus thus allows gene expression to be regulated by post-transcriptional mechanisms, such as alternative splicing. By limiting the access of proteins to the genetic material, the nuclear envelope also provides novel opportunities for the control of gene expression at the level of transcription. For example, the expression of some eukaryotic genes is controlled by the regulated transport of transcription factors from the cytoplasm into the nucleus, a form of transcriptional regulation unavailable to prokaryotes. The separation of the genome from the site of mRNA translation thus plays a central role in eukaryotic gene expression. The nuclear envelope and traffic between the nucleus and cytoplasm. The nuclear envelope separates the contents of the nucleus from the cytoplasm and provides the structural framework of the nucleus. The nuclear membranes, acting as barriers that prevent the free passage of molecules between the nucleus and the cytoplasm, maintain the nucleus as a distinct biochemical compartment. The weld channels through the nuclear envelope are provided by the nuclear pore complexes, which allow the regulated exchange of molecules between the nucleus and cytoplasm. The traffic of proteins and RNAs through the nuclear pore complexes not only establishes the internal composition of the nucleus, but also plays a critical role in regulating eukaryotic gene expression. Structure of the nuclear envelope. The nuclear envelope has a complex structure, consisting of two nuclear membranes, an underlying nuclear lamina, and nuclear pore complexes figure. The nucleus is surrounded by a system of two concentric membranes, called the inner and outer nuclear membranes. The outer nuclear membrane is continuous with the endoplasmic reticulum, so the space between the inner and outer nuclear membranes is directly connected with the lumen of the endoplasmic reticulum. In addition, the outer nuclear membrane is functionally similar to the membranes of the endoplasmic reticulum and has ribosomes bound to its cytoplasmic surface. In contrast, the inner nuclear membrane carries unique proteins that are specific to the nucleus. Critical function of the nuclear membranes is to act as a barrier that separates the contents of the nucleus from the cytoplasm. Like other cell membranes, the nuclear membranes are phospholipid bilayers, which are permeable only to small nonpolar molecules. Other molecules are unable to diffuse through the phospholipid bilayer. The inner and outer nuclear membranes are joined at nuclear pore complexes, the sole channels through which small polar molecules and macromolecules are able to travel through the nuclear envelope. Underlying the inner nuclear membrane is the nuclear lamina, a fibrous meshwork that provides structural support to the nucleus. The nuclear lamina is composed of one or more related proteins called lamins. In addition to providing structural support to the nucleus, the nuclear lamina is thought to serve as a site of chromatin attachment. The nuclear pore complex. The nuclear pore complex is the only channels through which small polar molecules, ions, and macromolecules, proteins and RNAs, are able to travel between the nucleus and the cytoplasm. The nuclear pore complex is an extremely large structure with a diameter of about 120 nanometers and an estimated molecular mass of approximately 125 million daltons, about 30 times the size of a ribosome. In vertebrates, the nuclear pore complex is composed of 50 to 100 different proteins. By controlling the traffic of molecules between the nucleus and cytoplasm, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. RNA that are synthesized in the nucleus must be efficiently exported to the cytoplasm, where they function in protein synthesis. Conversely, proteins required for nuclear functions, e.g., transcription factors, must be transported into the proteins required for nuclear functions, e.g., transcription factors, must be transported into the nucleus from their sites of synthesis in the cytoplasm. In addition, many proteins shuttle continuously between the nucleus and the cytoplasm. The rapid traffic of proteins and RNAs through the nuclear pore complex thus determines the composition of the nucleus and plays a key role in gene expression. Depending on their size, molecules can travel through the nuclear pore complex by one of two different mechanisms. Figure. Small molecules and some proteins with molecular mass less than approximately 50 kD pass freely across the nuclear envelope in either direction, cytoplasm to nucleus or nucleus to cytoplasm. These molecules diffuse passively through open aqueous channels, estimated to have diameters of approximately 9 nanometers, in the nuclear pore complex. Most proteins and RNAs, however, are unable to pass through these open channels. These macromolecules pass through the nuclear pore complex by an active process in which appropriate proteins and RNAs are recognized and selectively transported in only one direction, nucleus to cytoplasm or cytoplasm to nucleus. The traffic of molecules occurs through regulated channels in the nuclear pore complex that, in response to appropriate signals, can open to a diameter of more than 25 nanometers, a size sufficient to accommodate large ribonucleoprotein complexes, such as ribosomal subunits. It is through regulated channels that nuclear proteins are selectively imported from the cytoplasm to the nucleus, while RNAs are exported from the nucleus to the cytoplasm. Nuclear pore complexes by electron microscopy reveals a structure with eightfold symmetry organized around a large central channel, which is the route through which proteins and RNAs cross the nuclear envelope. Detailed structural studies, including computer-based image analysis, have led to the development of three-dimensional models of the nuclear pore complex. Studies indicate that the nuclear pore complex consists of an assembly of 8 spokes arranged around a central channel. The spokes are connected to rings at the nuclear and cytoplasmic surfaces, and the spoke ring assembly is anchored within the nuclear envelope at sites of fusion between the inner and outer nuclear membranes. Protein filaments extend from both the cytoplasmic and nuclear rings, forming a distinct basket-like structure on the nuclear side. The central channel is approximately 40 nanometers in diameter, which is wide enough to accommodate the largest particles able to cross the nuclear envelope. It contains a structure called the central transporter, through which the active transport of macromolecules is thought to occur. Internal organization of the nucleus. The nucleus is more than a container in which chromatin, RNAs, and nuclear proteins move freely in aqueous solution. Instead, the nucleus appears to have an internal structure that organizes the genetic material and localizes some nuclear functions to discrete sites. The aspect of the internal organization of the nucleus is the nucleolus, which, as discussed in the following section, is the site at which the rRNA genes are transcribed and ribosomal subunits are assembled. Details of internal nuclear structure are suggested by the organization of chromosomes and by the potential localization of functions such as DNA replication and pre-mRNA processing to distinct nuclear domains. The nucleolus. The most prominent substructure within the nucleus is the nucleolus, which is the site of rRNA transcription and processing, and of ribosome assembly. As discussed in the preceding chapter, cells require large numbers of ribosomes to meet their needs for protein synthesis. Actively growing mammalian cells, for example, contain 5 million to 10 million ribosomes that must be synthesized each time the cell divides. The nucleolus is a ribosome production factory, designed to fulfill the need for large-scale production of rRNAs and assembly of the ribosomal subunits. The nucleus during mitosis. A unique feature of the nucleus is that it disassembles and reforms each time most cells divide. At the beginning of mitosis, the chromosomes condense, the nucleolus disappears, and the nuclear envelope breaks down, resulting in the release of most of the contents of the nucleus into the cytoplasm. In meiosis, the process is reversed, the chromosomes decondense, and nuclear envelopes reform around the separated sets of daughter chromosomes. The process is controlled largely by reversible phosphorylation and dephosphorylation of nuclear proteins resulting from the action of the CDC2 protein kinase, which is a critical regulator of mitosis in all eukaryotic cells. B3. Mitochondria. Mitochondria play a critical role in the generation of metabolic energy in eukaryotic cells. Therefore, most of the useful energy derived from the breakdown of carbohydrates and fatty acids, which is converted to ATP by the process of oxidative phosphorylation. Most mitochondrial proteins are translated on free cytosolic ribosomes and imported into the organelle by specific targeting signals. Mitochondria are unique among the cytoplasmic organelles in that they contain their own DNA, which encodes tRNAs, rRNAs, and some mitochondrial proteins. The assembly of mitochondria thus involves proteins encoded by their own genomes and translated within the organelle, as well as proteins encoded by the nuclear genome and imported from the cytosol. Organization and function of mitochondria. Mitochondria are surrounded by a double membrane system, consisting of inner and outer mitochondrial membranes separated by an intermembrane space. Figure. The inner membrane forms numerous folds, cristae, which extend into the interior, or matrix, of the organelle. Each of these components plays distinct functional roles, with the matrix and inner membrane representing the major working compartments of mitochondria. The matrix contains the mitochondrial genetic system as well as the enzymes responsible for the central reactions of oxidative metabolism. The oxidative breakdown of glucose and fatty acids is the principal source of metabolic energy in animal cells. The initial stages of glucose metabolism, glycolysis, occur in the cytosol, where glucose is converted to pyruvate. Pyruvate is then transported into mitochondria, where its complete oxidation to CO2 yields the bulk of usable energy, ATP, obtained from glucose metabolism. This involves the initial oxidation of pyruvate to acetyl-CoA, which is then broken down to CO2